Today I'm going to talk a bit about my tool Phold, which uses protein structure information to improve the annotation of phages. So for some background, all the existing tools in this field, or most of them at least, use what's called sequence-based homology. I'm sure most of the listeners will be familiar with this. The way it works is pretty simple. You get your predicted proteins, you get the sequence, and you compare them to a database of proteins where we think we know the functions. The issue with this is that if you do this, most phage proteins remain unannotated. And I'm just going to put a quick little plug if you want to know more about this background to my colleague Susie's recent review published a few weeks ago. So there are many tools that have done this, but one of them is my own tool, Phold. So I'm selfishly going to talk a little bit more about that as an example of how this works. So on the right-hand side is the workflow. You start at the top with your phage genome. You predict the genes. You then use tools like hidden Markov models or MMSeq2 or BLAST, which are traditional sequence-based alignment approaches to compare your proteins to a database of proteins. If there are similarities, you propagate the functional label, and then you do a bunch of data cleaning and get nice outputs that everyone can upload and read and use and things like that. This tool, I'm a bit biased, but it's pretty good. But the big issue with it is lots of proteins remain unannotated. So ideally, we could use protein structures, not sequence, because there's more information in protein structures and they're also more conserved. Thanks to MVIF42 and other such tools, thankfully, we can now predict protein structures quickly on a computer directly from their sequence. The bottleneck was in comparing them, but my colleagues, led by Martin Steiniger and his group in Korea, developed a genius tool called Phold that makes comparing structures fast. The way it works is it takes a protein's three-dimensional structure in the middle, you can see, and then uses some fancy methods to convert this to a one-dimensional alphabet you can see on the right called a 3di string, which is one-dimensional, and it looks like an amino acid string. And thankfully, bioinformaticians have spent the last 50 years figuring out how to compare these really fast. The only bottleneck, though, with using this approach for phage or any annotation is the red box. MVIF42 is revolutionary, Nobel Prize winning, but it still takes quite a lot of time to run and needs big computers, especially if you do more than a few proteins. So my colleague Michael, in the bottom left, developed a protein language model, PROST5, that is thousands of times faster, and it's genius because it goes directly from amino acid straight to the 3di, which then you can use with Phold really fast. Phold, which is my tool, leverages these two tools to great effect. And so from left to right, the way it works is you get your phage genome, you can optionally annotate it with Phold. You take the proteins, you run the language model to predict the 3di strings. If you do have the structures, you can still use them here. Then it runs Phold to do structural comparisons against a big database of phage protein structures we've put together, and then it does summaries and plots and whatever other information you want. So a little bit more at the databases. We have more than 1.36 million. They're all very high quality. They come from phages and metagenomic proteins and other sources. They're freely available to download on Zenodo. And there's a nice website my co-supervisor Rob has put together that visualizes some of them. So please go check that out. How quickly can you run Phold? If you have a GPU, it's super fast, runs in minutes or seconds. If you don't have a GPU and you only have a laptop, you can run a phage or two in only a minute or two. And if you have a Mac, it can be a lot faster with the GPU on the Mac. If you're allergic to coding completely, you can also run it without coding using Google Colab Notebook that you can see with the QR code here. So all of this is all well and good, but how does Phold actually do? So Phold does pretty well. It can annotate above 50% of the proteins on most phages. So this is a data set of, I would say, the easiest phages to annotate. These are mostly cultured phages from infrared. Bouras gets around 40% of the annotations you can see, and then Phold gets around 55%, depending on if you use the language model or structures. I'm not really an expert in archaeal viruses, but Phold does pretty well. On average, about 40%, which I've been told is about as good as it gets for archaeal viruses too. So if you have them, give it a go. But I would say Phold is the best on metagenomic phages because of the sensitivity that structural comparisons provide. For example, on some Tara Oceans phages, Feroca can get around 20% to 25%, Phold gets above 50%. And then even on crass phages, which are super hard to annotate, and I'm sure Danica, who's following me, will talk more about this, Phold does pretty well, so around the 40%-ish mark compared to under 20% from Feroca and sequence-based methods. So to conclude, Phold is way more sensitive than existing sequence homology-based approaches. The language model removes the structure prediction bottleneck, and you can annotate more than 50% of proteins on your average phage. So this is just a QR code to the GitHub. Check it out, give it a go, hassle me, make issues, break it and test it, and hopefully it works well for you. And then I just want to acknowledge everyone who helped me with this work. Thank you, everyone.